Hello everyone, our project is called ErythroSight. The main purpose of our project is to detect sickle cell disease frugally. Some of the members in our international cohort include Sana, Kiran, Divyanshi, Rishika, Pranav, and myself, Toma. Sickle cell disease is the most common inherited blood disorder. It causes red blood cells to form rigid sickle-shaped cells, which block blood vessels, causing pain crises, organ damage, or death. It is estimated that in the next 30 years, 15 million children will be born with sickle cell disease. Unfortunately, the burden of the disease is highest in low-income countries, where mortality rates are high due to the lack of screening and treatment options. Sickle cell disease has a global prevalence, especially in many rural communities. The solutions we will discuss are focused on rural and remote communities of Nepal and India. These areas often have the highest disease prevalence due to the lack of screening and awareness of the disease. A high proportion of Nepal and India constitute rural communities, and the true prevalence of sickle cell disease in these communities is unknown. Some estimates indicate that the prevalence of the disease in certain communities in Nepal can be up to 25%, and in India up to 40%. Due to the high number of people, the project can also focus on a subpopulation, such as newborns or people within the reproductive age range. Currently, the sickling test is one of the most common techniques used to detect sickle cell disease in rural communities. Blood is mixed with a reagent that induces sickling of red blood cells in people with the disease. This is then observed under a microscope by a healthcare worker. Another common technique is the solubility test, where blood is mixed with a reagent that turns turbid if sickle hemoglobin is present, or turns clear if not. For both the sickling test and the solubility tests, a positive result indicates either sickle cell disease or sickle cell trait, and does not distinguish between the two. Recently, 2 lateral flow assays have been developed that can detect sickle cell hemoglobin and also detect hemoglobin C, which is common in African populations. While promising, these are more costly than the sickling and the solubility tests. A portable electrophoresis machine called Gazelle can also detect sickle cell disease and some other hemoglobin disorders such as beta thalassemia. The gold standard test is HPLC, or high performance liquid chromatography.
This slide show the sickling and solubility are the most cost-effective screening techniques currently available, but they lack in performance since they cannot distinguish sickle cell disease from the sickle cell trend, which would then require confirmatory tests. The lateral flow assays, that is the hemotype SC and the sickle scan and Gazelle, can distinguish the cases and have a high accuracy, but they are not as cost effective. The gold standard is expensive and usually not available or accessible in rural and remote communities. Next slides show us some example of the health post in rural Nepal, where microscope are available for performing the sickling test. However, the gold standard test, that is the HPLC, can be at least 150 km or 3 hours away, making the confirmatory test for diagnosing the sickle cell a real challenge.
Our proposed options for frugal detection of sickle cell disease can be broadly divided into optical and non-optical techniques. Optical techniques involve imaging blood microscopically, such as observing the cells, or macroscopically, so observing dry patterns, and other techniques related to optical scattering. Some non-optical techniques can be related to agglutination, capillarity, or viscosity of blood.
In our preliminary experiments, we imaged red blood cells using two low-cost microscopes, the Foldscope, which costs around $1, and Dipple, which costs around $100. These microscopes are more portable and cost-effective than conventional microscopes and can be paired with smartphone cameras for imaging. Some images from our preliminary experiments are shown here. Blood samples were mixed with a reagent, which was 2% sodium metabisulfite, then placed on a microscope slide, covered with a cover slip, and sealed. These images were taken around 1.5 hours after sample preparation using the Dipple black lens, which has a resolution of around 0.7 micrometer. The two images on the left are for a person with sickle cell trait. In the zoomed in image, we can see a few cells that are sickled, some of which are circled in red, while the rest of the cells are normal, so round and biconcave, some of which are circled in orange. This is a time lapse played at 120 times speed. Most of the cells are not sickled and are round and biconcave, while some of the cells are sickled. The cells here are moving only because the seal was broken on the right side of the cover slip because the lens housing was too close to the slide. Some images from a conventional microscope at the hospital are shown. Note that these images do not have the spherical aberration that was seen in previous images. If the goal is to just detect sickle hemoglobin without distinguishing between sickle cell trait and sickle cell disease, like how it is currently being done in the field, the estimated number of cells required for imaging could be in the order of 100 to a few thousands. So only a couple of fields of view would be sufficient for imaging, and this could potentially be done with a conventional microscope or a low cost one, like we have done in our preliminary test. However, we could possibly distinguish the different cases by also analyzing the shape of the red blood cells, for which there has been some recent evidence. The exact number needs to be determined empirically, but we estimate that around 10,000 to a million cells might be needed to accurately distinguish the different cases based on shape alone. More work needs to be done for this, but uh, a microscope with automated scanning and disease detection is suitable for this purpose. Looking at the cost performance plot, an automated sickling test would cost similar to the traditional sickling test, but would have a much higher performance and would potentially be able to distinguish sickle cell trait, sickle cell disease, and healthy cases automatically without the need for a highly skilled personnel, which would be ideal for screening the disease in rural or remote communities. Moving on from microscopic imaging to macroscopic imaging, we looked at drying patterns of blood. In this experiment, the mixture of blood plus reagent was placed on a microscope slide and dried under ambient conditions. The preliminary results were quite exciting. There seemed to be differences in the drying patterns for the cases of sickle cell trait and normal blood. There were differences in the thicknesses of the band, the outer bands, the overall intensities of the image, uh, potentially due to differences in the migration of cells to the contact line during evaporation. These images were taken from a 2x objective, and the diameter of each spot is roughly around 5 millimeters.
Another direction to explore and work on can be going back to the basics of class 12 physics and observing the behavior of how RBCs with sickle cell traits behave differently from normal ones when a beam of light interacts with them. The literature by bar shows that the scattering pattern of the RBCs with sickle cell traits is quite different from the healthy and normal red blood cells. Various biochemical properties too can be reduced from the dynamic light scattering. The literature concludes that the mechanical properties vary as per the morphology of sickle cell RBCs. Hence, if we find a frugal way to measure the mechanical parameters like let's say viscosity, then they also can be used as a way to detect sickle cell anemia cases. Further, this technique in combination with other optical imaging techniques can also be used to study and analyze other diseases related to RBCs.
Coming to the next slide is the agglutination. So basically in serological analysis in hospitals, this is one of the common experiment which is being carried out, where an antigen is being mixed with an antibody. There is solubilization, there is agglutination, there is precipitation. So in the figure you can see the RBCs are diluted as 1 is to 1, 1 is to 2 till 1 is to 28. and there is some agglutination agent, maybe it is treated with antibody. This is the same principle we want to add over here. We want to take the healthy RBC as well as the anemic RBC, and we want to treat with some of the agglutination agent, and we want to see the differences which can be observed using naked eyes. This experiment can be done using microtiter plates or a cavity slide, and we can really observe that is there any changes, in the agglutination pattern observed or not. Recently there has been a new approach of using nanoparticles and seeing the interaction of RBCs with nanoparticles. In the figure, these are the RBCs which are treated with the n number of nanoparticles, and this is the SIM, that is scanning electron microscopic images, and how they interact. There are nanoparticles like which are incubated with titanium dioxide, zinc nanoparticles, polymeric nanoparticles. In the recent trend, when nanobiotechnology is being accessible, we really looking forward to have one of the agglutination agent as nanoparticles, and we really need a lot of literature review. We really need to study well whether what's the interaction of RBC with nanoparticles, and if one of the nanoparticle, for example silver nanoparticles, can act as agglutination agent. So can silver nanoparticles, which can be extracted easily from using plants, is there any differences, or is there any difference in agglutination pattern when we treat silver nanoparticles or any nanoparticles with the healthy RBC and the sickle cell anemic RBC? Of course, while doing this experiment, our main approach is to get frugality, and it should, we need to be reproducible, we need to have maximum specificity and sensitivity, and we need to eradicate false positive and false negative tests. So this is the thing that we are looking forward to.
So, the conclusion and the future work of this project is that overall, we believe our proposed solutions are of course similar to current screening techniques and potentially have superior performance. These alternatives can be suitable for screening sickle cell disease in rural and remote communities. We hope to explore many of the proposed solutions in details in the coming months. Thank you for listening. Please visit our Notion page for more information. We want to thank Professor Manu Prakash, Taylor, Anton, and Subir for the help throughout our project.